This is the first of a new type of journal club that Bob and Pete are going to be doing together.
Why we chose this paper and how we got here. In popular nutrition culture, people are starting to think of metabolic flexibility in terms of whether they're burning carbs or fat. And we'll get into like more nuanced detail and like what those differences are with this study, and we'll riff off that and talk about some of the more nuanced pieces. But in essence, yeah, when we talk about metabolic flexibility, whether or not the body has the ability to both utilize carbs and fat as fuel, and why we use it as a model in the MedFix Foundations course is because what we're seeing in the research is that your ability to utilize fat as a fuel will determine or potentially predict your health outcomes. And that happens off a wide range of people.
This basically says we already conducted this study. This is another paper. So this is the 2019 paper where they talked about performance, which you'll often find too in studies is it'll be a follow-up study. You won't know it. There'll just be one line and it says, we described this previously. And then there's just a number or a citation and you think, then you go down the rabbit hole, you go to that citation, and oftentimes that citation cites another citation. And then eventually, hopefully, you get to the original protocol or study where they get into the design and methodology and the baseline characteristics and all that stuff. And that's what the 2019 study has in this case. They took the approach too as well. This isn't necessarily what you have to do in science. It's kind of like a misunderstanding of the quote unquote null hypothesis. Because it's the null hypothesis, they based it off of the paradigm and the conventional belief that low carb, high fat diets are bad news if you're going to do a 5K time trial, because that means you're going to have to exercise at high intensity. That in the 2019 paper, there was an impairment during those 5K runs. Carbohydrate is effectively the preferred fuel, the principal energy fuel, the fuel you need to actually drive that performance. How do they figure out how many carbs am I burning? How much fat am I burning? What's my VO2 max? And it turns out that it also has to do with the crossover concept. And it also has to do with metabolic flexibility, at least the classical definition. Not only what your percentage of carbs you're burning and fat, but what is the rate of carbohydrate and fat utilization, which is really important. A test of metabolic flexibility, or if we're looking at RQ, we're also looking at mitochondrial function, because the TCA cycle and especially during exercise.
Basically, we're just making the assumption that any measure between 0.7 and 1 is just some blend of the two.
Cells are not utilizing one or the other, is that they can utilize both. They can utilize fat and glucose. There's something called a Randle cycle, and that in some ways it argues, it says, nope, one bullies the other. And so if you're burning fat, you can't burn carbs. I would just say like, it's not how it works. Because if you imagine like, if you're just burning straight carbs, right? And then you're going to transition to burning fat. Do you think like it's a switch and all of a sudden the cell goes from burning carbs to burning fat? Or is it burning a mix and it's predominantly burning one or the other? I think it's the latter, because if it's not the latter, I don't think we would be here. You want to get the person to max in like say 15 to 18 minutes.
Yeah, there's a sweet spot.
Yeah. All of this stuff is based off of that CO2 and VO2 on the left-hand side. And then you have that RER, which we talked about, that RQ or RER. And then based on that, you can look at absolute rates instead of talking about these percentages.
When you're going to see a crossover point, there's some people that publish crossover points with absolute measurements for the carbohydrate utilization and fat oxidation. That definition points to the crossover point being it's a ratio. So it's a percentage. So I would take that as 50% or more. Like 51%.
If you look at the carbohydrate oxidation at rest, it's about 30%, 30 to 40%. And so this is saying you're almost 50-50 before you even hop on the treadmill.
Those are the predictions leading into this study. Those are predictions based on old concepts and older research, right? Of mixed diet individuals per se.
Yeah, well, old as in probably people who are not looking at it. Yeah, it's
right.
There's people, there's like the low carb guys, there's the high carb guys. So metabolic flexibility, this is another term that it depends on the paper that you read or the person that you ask. But generally, like if it's a broad term, it's the adaptation of fuel oxidation to a given change in fuel availability. It's the gold standard for testing insulin resistance because they fuse insulin and they give you a fixed concentration of glucose. They look at how much glucose you're taking up. In some ways it makes sense. You're going to look at glucose, although actually it doesn't make sense to me. Because I think fat that's our fuel. They use those two things. So they actually have 2 data points, right? They talk about what does it look like in fasting conditions? And then what does it look like under a stimulus? Like if you want to read about metabolic flexibility, you're probably reading this guy Galgani. And it's funny because they're like metabolic flexibility of glucose is suspect. We should probably be looking at lipids. And they say it as if they weren't themselves the ones that did all that research on glucose, but they did, which is fine. Like you move on, but they make it seem like, those rascals. And I'm thinking like, you're on that paper. It's you. So what he's saying is they're oxidizing the fuel. It's just the cells, it's not available to the cells. But to me, that's another way of saying there's insulin resistance, meaning that there's something that is resisting the glucose in the cells. And so their uptake is less. I think that's also, it's highly relevant, but they are saying, try not to confuse those two, but it does make this more confusing. Glucose versus fat. So most studies that are looking at metabolic flexibility, they look at glucose and they do them with hyperinsulinemic euglycemic clamps. This is Fernandez Verdejo. The other author is Galgani. So they're like, curiously, like, why is it curious? Explain yourselves. But anyway, it's the same thing with insulin resistance. All we look at is glucose. And to me, it looks like if there's a nutrient overload in the cell, if the cell is overloaded, like if it's basically getting more fat at rest or whatever, that's fuel. And so when you give that cell glucose and it already has what's called ectopic fat, some people call it lipotoxicity, for whatever reason, is that fat starts spilling over into other cells, including skeletal muscle. And when you give it glucose, sometimes it's already full or it's partially full. When Galgani talked about fuel availability and it says, wow, these type 2 diabetics, they're not taking up as much glucose. That is completely, to me, it's completely physiological, even though it's not a state you want to be in. Because when you talk about the human body and we say there's 37 trillion cells, which is, it's got to be an estimate. It's hard to count that number. Is that when you look at your cells, like what you're made of, it's something like 90%. 90% of your cells are red blood cells. And those red blood cells don't have mitochondria and they don't have a nucleus. And those cells basically have the glycolysis path, and that's it. And so those red blood cells, they'll rely on glucose. So you're probably sparing in some ways too, that you're making sure that your red blood cells are functioning. So of course, what are they doing too? They're carrying oxygen, hemoglobin, and they're carrying substrates. They're super important. I always find it interesting that
they're just looking for a different cause for that metabolic inflexibility.
Yeah.
What you're saying.
Yeah. Maybe we're not oxidizing it. Like we're impaired in our ability to oxidize it. And if that's the case, then it's going to accumulate. The deposition is going to exceed the mobilization and oxidation because we've got this defect that biases it towards storage. My bias is insulin was and is absolutely critical and to the function of a cell and to nutrient uptake and partitioning. When oxygen came on the scene, atmospheric oxygen, along with it came ROS. And so it's oxygen/ROS. We're talking about 4 billion years ago. If you look at the oxygen, you can see like a big increase. And this is the great oxidation event where a lot more oxygen came on the scene closer to what we see now. So it's not to say that insulin or leptin are not important. They absolutely are. But you could argue that they're bolt-ons onto ROS as the underlying mechanism that allowed cells to survive. Reactive oxygen species are fundamental and they're physiological. They can be pathological, but I would say that what has been underappreciated is a physiological role of ROS.
These ideas of ROS that we're going to get into, they almost seem so far out of left field for the simple fact that they're not part of like the pop culture nutrition science, but they kind of are with the whole obsession with antioxidants. Like you're just talking about, the purpose of the antioxidant is to quench that reactive oxygen species or that free radicals that it doesn't cause damage to the cell. But it's just funny to me how the antioxidants became such a part of like, pop nutrition culture, but then they kind of like missed the point. They stopped worrying about it or exploring it after that.
Yeah, that's a good idea. It reveals the bias or the dogma in the field that reactive oxygen species are bad, so antioxidants must be good. Because if you start messing around by just taking lots of antioxidants, you're messing with that physiological signaling. The point is here, we'll compare it to reactive oxygen species, where these have 12 valence electrons, and it's not counting those 4, but it would have 8 and 8 is 16 with the 2 oxygen molecules. And then superoxide is that it's got an extra electron. So it actually has 13 around the shell. And on the lower right, you can see that.